In 1954, British chemist G. A. Snow and colleagues isolated a compound from Mycobacterium flae, which served as a growth factor for MAP. The isolated substance is called mycobactin, and mycobactin enables mycobacteria to chelate, grab and steal iron from its host. When isolating the mycobactin, the researchers used aluminum oxide and noted the experiment resulted in the direct isolation of crude crystalline aluminum mycobactin from dried Mycobacterium flae. Snow and colleagues observed the aluminum atom is firmly held by the normally iron-chelating mycobactin. After examination, 15 cattle with nodules in the connective tissue underneath their skin were shown to have aluminum hydroxide granulomas related to vaccination. Aluminum hydroxide is a common adjuvant in vaccines. Biopsy specimens revealed mycobacteria in 8 of 14 granuloma. Mice were immunized with 5 experimental subunit vaccines based on secreted antigens from Mycobacterium. The list included incomplete Freund's adjuvant and aluminum hydroxide. The mice were then infected with Mycobacterium tuberculosis, and incomplete Freund's adjuvant was found to be an effective TB subunit vaccine. Aluminum hydroxide, on the other hand, increased susceptibility of the animals to a subsequent TB challenge. Strikingly, the use of aluminum hydroxide as an adjuvant primed an immune response which increased the susceptibility of the animals to virulent challenge. Complete Freund's adjuvant consists of inactivated and dried Mycobacterium tuberculosis in oil emulsion. It is used as an immunopotentiator in animal models of autoimmune diseases. Incomplete Freund's adjuvant lacks Mycobacterium tuberculosis. The neuroprotective potential of vaccines containing either tyrosine hydroxylase or glutarimer acetate in complete Freund's adjuvant was tested in the MPTP mouse model of Parkinson's disease. Surprisingly, we observed that the main beneficial factor in these vaccines was the complete Freund's adjuvant. Complete Freund's adjuvant vaccination promotes neurorestoration. Granulomatous enteritis, a disorder with many similarities to human Crohn's disease, were found in a cluster of 6 horse fatalities. When tissues were examined, researchers unexpectedly found the presence of excess aluminum concentrated within microorganisms in the intestinal wall. The mycobacteria responsible for intestinal infection in horses is Mycobacterium avium, and not MAP. It is thought that MAP evolved from Mycobacterium avium. In the article Bacterial-Metal Interactions, the potential role of aluminum and other trace elements in the etiology of Crohn's disease, the authors suggest that bacteria colonizing the intestinal wall of patients with Crohn's disease incorporate aluminum and other trace elements through their iron uptake system, and that as a result of the adjuvant properties of aluminum, they induce an exuberant granulomatous inflammatory response. Over a period of 8 years and at a cost of £1.5 million, a research team at St. George's University of London has developed a state-of-the-art modern therapeutic vaccine to kill MAP and get rid of the distressing symptoms that blight the lives of so many sufferers and their families. Methotrexate, azathioprine, 6-mercaptopurine, 5-aminosalicylic acid, cyclosporine A, rapamycin, and tacrolimus are all drugs that have been used to treat Crohn's disease. Methotrexate, azathioprine, 6-mercaptopurine, 5-aminosalicylic acid, cyclosporine A, rapamycin, and tacrolimus have been shown to inhibit MAP growth in culture, indicating that MAP infections may have been treated for decades. The authors of On the Effect of Thalidomide on Mycobacterium avium subspecies Paratuberculosis in Culture state, HPD is a component of thalidomide and has been shown to inhibit MAP growth. These data are compatible with the hypothesis that thalidomide, like other anti-inflammatories and immunomodulators, may act in part as an anti-MAP antibiotic. In a mouse model of Parkinson's disease, thalidomide improved the neurotoxicity induced by MPTP, as seen by a significant raise of striatal contents of dopamine and simultaneous decrease of monoamine oxidase B, MAOB. Our results suggest that thalidomide could be investigated as potential adjuvant therapy for Parkinson's disease. University of Central Florida Research Foundation developed a patented protected test for MAP, and Red Hill Biopharma has acquired the exclusive license to the test. RHB-104 is a combination antibiotic therapy in oral pill form, with intracellular antimycobacterial and anti-inflammatory properties. In a clinical trial, RHB-104 placed 8 of 10 pediatric Crohn's patients into remission. Dr. Barodi, developer of RHB-104. Crohn's is a more difficult disease to treat because bacterial infection develops very slowly. The bacteria involved are the most slowly dividing bacteria known to man, so medications have to be taken for a long time. Fecal microbiota transplant is a procedure in which fecal matter is collected from a tested donor and placed in a patient by colonoscopy, endoscopy, sigmoidoscopy, or enema. Dr. Barodi cured a nurse with Crohn's after fecal transplant. Today, nurse Susan Heskett is full of energy and walks several kilometers each day, but 13 years ago she was very ill, diagnosed with Crohn's, a disease she believed would never be cured. Professor Barodi also claims symptoms of Parkinson's eased in 3 patients treated for chronic constipation, some to the point that the Parkinson's could not be detected by other neurologists. Constipation is a symptom of Parkinson's disease that can precede the development of other symptoms by 10 years. Dr. Barodi treated a 74-year-old man with a 4-year history of constipation with the antibiotics vancomycin and metronidazole. At 21 days, he reported his constipation to be markedly improved, defecation with ease and no bloating. He also noticed a dramatic improvement in his Parkinson's symptoms with decrease in tremor commencing from about 10 days into therapy. Vancomycin originated from Nocardia orientalis and may be acting in the gut flora to suppress a related pathogen's release of its toxins and may help uncover the riddle of Parkinson's disease. Gastrointestinal dysfunction in Parkinson's disease. Gastrointestinal motility is frequently disturbed in Parkinson's disease, manifesting chiefly as dysphagia, impaired gastric emptying, and constipation. The relationship between the nervous system and therapeutics in pulmonary tuberculosis, from 1916. There is a decreased motility on the part of the gastrointestinal tract, leading to stasis and constipation. There is slower action of the nerves supplying the gastrointestinal tract in people suffering from Parkinson's disease, so that movement of food during the process of digestion is slowed down. Food remaining in the stomach for long causes acid and gas buildup. High prevalence of gastroesophageal reflux disease in Parkinson's disease, a questionnaire-based study. Increased risk of pulmonary tuberculosis in patients with gastroesophageal reflux disease. Analogous to the tuberculoid and lepromatous forms of leprosy, Crohn's disease may have two clinical manifestations. One is aggressive and fistulizing, perforating, and the other is contained, indolent, and obstructive, non-perforating. An open trial on 5 patients with Crohn's colitis or ileocolitis were given the leprosy antibiotic dapsone, and the therapy was apparently effective in 2 out of 5 patients. In the 2 responders, antibody levels to a soluble extract of M. Paratuberculosis were significantly greater than in the 3 non-responders. There was a rise of 39% in antibody titers following the treatment. This rise in antibody levels is similar to that observed after treatment of tuberculosis. Our data suggest that a mycobacterial species or another pathogen that cross-react with MAP, sensitive to dapsone, may in a subset of cases be responsible for the development of Crohn's disease. Dr. Marcel Baer is a full professor of medicine at McGill University in Montreal. Dr. Baer's lab uses bacterial genetics to study the epidemiology and pathogenesis of mycobacterial diseases. Dr. Baer has developed a mouse model of Crohn's disease. Mice are infected with MAP and are treated with combinations of antibiotics to determine their efficacy in Crohn's disease therapy. Mycobacterium paratuberculosis has been found within Crohn's disease tissue, but there has been controversy concerning its role in the disease. We have now shown that these mycobacteria release a complex molecule containing a sugar called mannose. This molecule prevents a type of white blood cell called macrophages from killing internalized E. coli. Scientists have previously shown that people with Crohn's disease have increased numbers of a sticky type of E. coli and weakened ability to fight off intestinal bacteria. The suppressive effect of the mycobacterial molecule on this type of white blood cell suggests it is a likely mechanism for weakening the body's defense against the bacteria. Humans are exposed to MAP by eating ground beef or cuts of beef, by drinking raw or pasteurized milk, or by drinking groundwater from MAP-contaminated farms or exposed water supplies. Researchers from University of Lancaster in England reported exposure to paratuberculosis may include inhalation of the pathogen from either showers or the spray of waterfalls and rivers infected with MAP. Inhalation has been shown as a route for the infection of cattle, and lung involvement is well described in adults with Crohn's disease. The disease in children often begins with a cough and a mild inflammation of the throat and lungs. In Japan, 16 Crohn's patients already in remission were placed on a diet of 1 serving of fish and 1 serving of meat per week, and after 2 years, 15 of 16 stayed in remission. A second group of 6 Crohn's patients ate whatever they wanted, and after 2 years, only 2 were still in remission. There is no question that Japanese people are eating a lot more fats and saturated fats than they used to, said Craig Wilcox, a professor at Okinawa International University. They also have a lot more meat in their diet, a higher proportion of milk products that were not really available previously, and more bread. In the early 20th century, American milk supplies were at times heavily contaminated by M. tuberculosis and M. bovis. As a result, mandatory milk pasteurization was instituted to kill the pathogens. Australian researchers were able to culture MAP from milk pasteurized at 179.6 degrees Fahrenheit for 15 seconds. American milk is pasteurized at 161.06 degrees Fahrenheit for 15 seconds. In 1998, Northern Ireland health officials found nearly 1/5 of the Irish milk supply was contaminated with MAP. This led to an English survey of their pasteurized milk supply, and they found 3 out of 100 milk cartons grew live MAP bacteria. Each quart had to contain at least about 1 million paratuberculosis germs to come up positive. In the Czech Republic, 51 samples of retail powdered infant formula, produced by 10 companies from 7 European countries, were tested for MAP. MAP, in concentrations from 48 to 32,500 cells per gram, of powdered infant milk were found in 18 of 51 investigated samples, or 35% in this study. More than 10,000 cells per gram were present in 4 samples. Such concentrations mean that one package of milk contains 5 million MAP cells, which are ingested by a bottle-fed baby over the course of several days. Premature babies and bottle-fed newborns can be affected by pro-inflammatory triggers from a huge number of mycobacteria, despite not suffering from infection with bacteria or viruses often linked with the etiology of Crohn's disease. The hypothesis that MAP and perhaps other mycobacteria in baby formula play the role of the missing environmental factor in the etiology of Crohn's disease is supported by many papers which describe breastfeeding as a protective factor against Crohn's disease. In the United States, 702 pints of retail whole milk were purchased, and viable MAP were cultured in 2.8% of the samples. More MAP-positive samples were identified during the third quarter of the year, July through September. Of the 22 brands of retail milk tested, 55% yielded at least one sample positive for viable MAP. In Cyprus, 225 bulk milk tank samples were tested for MAP using 2 different methods, traditional culture and a new bacteriophage test. After culturing 225 samples, MAP was cultured 2 times. Using the B29 bacteriophage test, they identified 50 contaminated samples. In England, 56 milk samples were tested and no MAP were cultured. Using the bacteriophage test, 1 milk sample was positive for MAP. In Cyprus, nearly 97% of the samples had Mycobacteria species present. In England, 35% of the milk had Mycobacteria species present. A University of Virginia statistical analysis concluded that middle-aged men who are heavy milk drinkers, more than 16 ounces a day, are 2.3 times more likely to develop Parkinson's disease than non-milk drinkers. A meta-analysis concluded that dairy consumption is a risk factor for Parkinson's disease. Men increase their Parkinson's risk by 1.8 times and women 1.3 times. These data suggest that dairy consumption may increase the risk of Parkinson's disease, particularly in men. The antibacterial activity of 18 naturally occurring compounds were tested against 3 strains of MAP. 1 derived from a cow, 1 derived from raw milk, and 1 derived from a human being. The most effective compound was trans-cinnamaldehyde, which is a component of cinnamon tree bark. The chemical structures of trans-cinnamaldehyde show slight similarity to isoniazid and pyrazinamide, drugs shown to be effective against M. tuberculosis. These results indicate that trans-cinnamaldehyde has a neuroprotective effect on dopaminergic neurons, and that this effect may be associated with the inhibition of inflammatory responses. Neurological scientists at Rush University Medical Center have found that the spice cinnamon is metabolized into sodium benzoate, which enters the brain and thus stops the loss of the Parkin and DJ1 genes. Sodium benzoate protected neurons and normalizes neurotransmitter levels and improves motor functions in mice with Parkinson's. This article proposes that genetic defects associated with Parkinson's also result in a permissive environment for MAP infection, ineffective xenophagy. It postulates that beginning as an enteric infection, MAP, via the vagus nerve, initiates a pathologic process that results in a targeted neuroinvasion of the central nervous system. The article proposes that MAP infection and resultant Parkinson's pathology are due, in the genetically at-risk and age-dependent, to the consumptive exhaustion of the protein quality control systems. Is there a role for Mycobacterium avium subspecies Paratuberculosis in Parkinson's disease? We have observed the presence of MAP in Parkinson's disease patients by polymerase chain reaction, positivity to MAP3865C peptides,